Controlling when and where cells express a gene is a powerful technique in research and therapeutic fields, from controlling differentiation in small subsections of an organoid to restricting gene and cell therapy to target tissues and reducing off-site activity. Current techniques for expression regulation include exogenous application of chemicals, optogenetics, and sonogenetics. However, all of these techniques have limitations. For example, optogenetics requires the fitting of optical fibers into animals being studied, and even then, the penetrative power of light limits how deep within a tissue we can activate. Magnetogenetics is a technique using magnetic fields to regulate cellular activity. With our magnetogenetics system, MaGenTa, we seek to resolve current limitations and provide a solution with three-dimensional selectivity and high precision for a low price.
In this project, we employ three methods to modulate gene expression based on magnetic fields. Two of these make use of magnetic nanoparticles. These nanoparticles may be incredibly small at 10 to 20 nanometers, meaning that they generate heat under a high-frequency alternating magnetic field. Such particles can be mixed into the media and can switch on cellular thermosensitive pathways without the need for specific interactions with the cell membrane. The thermal pathways that we are using involve transcription factors that undergo a conformational change upon heat shock. TLPA and TCI, repressor proteins from Salmonella and bacteriophage lambda respectively, are proteins that dimerize and repress expression at temperatures ranging from room temperature to 37 degrees. Under heat shock, they monomerize and release repression of their targets. Alternatively, the nanoparticles may be slightly larger at 250 nanometers and coated with a nickel NTA coating that allows them to interact with his-tagged cell surface proteins. This means that when a low-frequency magnetic field is applied, they oscillate and pull or push on the cell membrane. This mechanical force can then activate internal pathways, such as the cell wall integrity pathway in yeast, which we activate using a form of WSC1, which has been engineered to extend out of the cell wall such that it has an extracellular his-tag, which can interact with nickel-coated nanoparticles in the media. When activated, WSC1 changes conformation and interacts with a guanine exchange factor. This switches on a small G protein and then activates a MAP kinase cascade. One of the transcription factors activated by this cascade is RLM1, and we are using a plasmid containing LAC-Z under the control of an RLM1 dependent promoter to induce a color change to blue under a magnetic field. The third way to make cells respond to magnetic fields is by employing the magnetosensitive protein EPG. This gene is found natively in glass catfish, which swim away from magnetic fields. It undergoes a conformational change when placed in a magnetic field, bringing its termini closer together. Thus, by fusing each half of a protein of interest to the different termini of EPG, it can be activated and regulated by magnetic fields. Since each of these pathways is activated primarily by a different type of magnetic field, we have designed a hardware system that activates two of these along different axes. By combining the two cellular responses into a genetic AND gate, we can then achieve precise spatiotemporal control, since only the point of intersection between the two axes will have both the necessary inputs.
From the start of our project, we have aimed to make sure that what we do remains relevant for the fields in which we want to innovate. After a literature review, we reached out to industry representatives and researchers to learn how this technology could be useful to them. Following their advice, we narrowed down our target applications to neuroscience, organoid research, and CAR T-cell therapy. We focused on making the specifications of the technology suitable to their application. For example, ensuring that the size and the material of the magnetic nanoparticles would be safe for use in a therapeutic environment, and the degree of precision needed for organoid research. We also spoke to firms in optogenetics and manufacturing to determine how potential users may interact with the technology, and we fed this back into our design cycle. Additionally, to make our hardware more accessible to users without an engineering background, we demoed it at a local event and received feedback from users, allowing us to improve the user interface.
Standard magnetogenetic hardware systems are expensive and inflexible, so we built our own. Initially, we sought hardware to characterize and better understand our controllable pathways. For two of our constructs, EPG and WSC1, we built an electromagnet test rig for producing high field strength, static, and low-frequency oscillating magnetic fields. For our thermal construct, we designed and built our own high-frequency coils based on induction heaters to heat our magnetic nanoparticles and activate the pathway. we were able to estimate the spatial distribution of temperature over time using heat diffusion models. Finite element methods were applied to each design to model the fields generated by these electromagnets, and we used the results to make iterative improvements. Learning from these initial devices, we then developed MagentaBOX, a research platform for 2D spatial targeting using magnetogenetics. MagentaBOX combines two different electromagnets on motorized stages with imaging capability for color, fluorescence, and temperature. This allows us to precisely move and power our electromagnets around our cells and to measure the spatial distribution of response over time. We showed our prototype to some potential future users and asked them what they thought. Based on their feedback, we built a graphical interface, user guide, and integrated our control electronics into the box to improve reliability. We hope MagentaBOX will be built, used, and improved on by future teams and researchers, so we have included links to a build guide and all the necessary CAD, laser cutter, and 3D print files on our wiki.
250nm magnetic nanoparticles can be viewed under a light microscope. Through simple incubation with cells, they can bind to the extended WISC-1 on our yeast through the interaction between nickel and haystack. The reporter plasmid we use has the LAX-A RM1 LAXAT reporter system. LAXAT is under the control of a GAL-LAX-A synthetic promoter, which is based on a normal GAL promoter, except the UAS is replaced by a LAX-A binding site. LAX-A is fused with RM1. When the mechanical sensor WISC-1 detects changes in osmotic pressure or mechanical stress, RM1 will be phosphorylated, and the fused LAX-A will activate the expression of LAXAT, which codes for beta-galactosidase. XGAL is used as an indicator for the activation of WISC-1. When XGAL is added to the activated colony, it will be cut by beta-galactosidase and form a blue precipitate, making the colony blue, while the non-activated colony remains white. WISC-1 can be activated by various forms of stress affecting the cell wall. For example, incubation at 37 degrees overnight, or long-term incubation at 30 degrees. When nanoparticles bind to WISC-1, they respond to external magnetic fields and vibrate to generate a mechanical force on WISC-1, which also activates it. After 1 hour of activation, we could see a slight difference in color between the activated group and the control group. As the activation takes around 1 hour, the expression of beta-galactosidase and cleaving of XGAL may take longer. Adjusting our protocol to optimize signal strength led to further trials showing that the color change will be more obvious after overnight incubation, and even more visible after an additional 2 days incubation at room temperature. The color change between activated and non-activated groups indicates the successful activation of WISC-1 by a magnetic field.
2 similar pathways were engineered in E. coli, involving the transcription factor TLPA and TCI. Each of these involved a pair of plasmids. The first plasmids constitutively express the sensor protein, TLPA or TCI, while the second plasmid contains a fluorescence reporter under the control of the sensor transcription factors. The reporter could be changed to other effector proteins, which allows integration into our AND gate system. The other pathway we engineered in E. coli was the electromagnetic perceptive gene, or EPG. For this, we set up 2 different systems. One of these is to characterize the behavior of EPG, while the other is to integrate it into the AND gate system. For characterization, we designed an EPG nanoluciferase fusion protein that could monitor EPG activity under different field strengths. On the other hand, for the integration into the AND gate system, we designed an EPG TAF protease that could cleave effector proteins. For the TCI pathway, we incubated cells transformed with both the relevant plasmid at various temperatures. The results were qualitative, and they matched our expectations. The next step will be to build on our quantitative test to obtain comprehensive data that could guide our computational models for TCI behavior. For the EPG nanolook system, we have carried out initial characterization with the plate reader. The EPG has been tested at 24 millitesla, 12 millitesla, and 6 millitesla using our magnetic device. The result shows an unexpected trend that applied magnetic field may reduce the activity of EPG nanolook. However, there are innate drawbacks of this method, since we cannot measure luminescence level with the magnetic field is being applied. To overcome this, we developed a new custom hardware to measure luminescence. Although the hardware functioned properly, the detector was not sensitive enough for EPG signal. We altered the expression promoter, reaction mix, and have proposed a more sensitive device. For the EPG TEF system, we carried out a computational method to optimize the sequence of the linker connecting EPG to the TEF domains. The next step will be to first characterize the basic linker system, and then screen construct with different linkers for optimal activity.
With the potential of this technology clear, we reached out to stakeholders to get a sense of the feasibility of our application ideas. The most significant of these are organoid research, using CAR T-cell therapy for solid tumours, and neuroscience research. Organoids are bundles of multipotent stem cells which self-arrange into an organ-like structure. However, unlike in an organism, their development must be guided by signalling molecules applied by a researcher. Three-dimensional control of expression of morphogens and transcription factors opens new avenues for experimentation and could accelerate drug trials with more consistent organoid structures for preclinical testing. CAR T-cell therapy is a form of immunotherapy where a patient's T-cells are genetically modified to express a chimeric antigen receptor that enables them to destroy cancer cells by targeting overexpressed antigens and triggering apoptosis pathways. Its current use in treating solid tumours is limited, but MaGenTa could provide a way to address this. A CAR T-cell could be bound to a number of magnetic nanoparticles before reintroduction to the body. Then, using a series of genetic AND gates, the T-cell would only be able to function in an area where both correct antigens and a set of overlapping magnetic fields are present, reducing off-site activity and improving recovery time. In neuroscience, optogenetic methods are commonly used in mouse models as they allow up to single neuron stimulation. However, experiments on this scale require expensive equipment, invasive surgery, and high levels of expertise. Surgeries on the brain can alter how it behaves, reducing the validity of studies. To solve this, magnetic nanoparticles and a viral vector could be applied intravenously to avoid surgery.
We hope MaGenTa is a technology which can be built on in the future using the data we provided. None of this would have been possible without the help and guidance of our instructors and the researchers we've contacted. So we want to say a big thank you to them and to our sponsors who made this project possible.